Hello everyone, this is a video on vulval precursor cell, how it develops from our organism C. elegans. Mostly it's beneficial for CSIR point of view. Those are preparing for the upcoming exam. So it's a scoring unit, I believe, developmental biology unit 5, up to 6 questions in from each unit, and it's having a weightage. From here also you receive around 6 questions in like CSIR and DBT comparatively less. Thus we have this topic. Today I will make you understand, I will try to make you understand about the vulval development, how the C. elegans, a hermaphrodite, and we all know C. elegans mostly called the like king of developmental biology. It's a model organism. Mostly C. elegans and Drosophila both are used as the model organism. It's a model also, and as I have using a mouse only for my pointer, so maybe it's not writing properly. But thing is, the C. elegans we will start from here. It's a nematode worm, mostly hermaphrodite. Hermaphrodite means at some stages of life it is a male, and at later stages, you may say at early stage of life it is a male, and at later stages it develops into a female. So at in each lifetime, in a lifetime, it's a male, and further it develops into a female. Thus at early stages of development, it is male, produces sperm, and it is stored. And at later stages, when they grow old, they become female, they develop ovaries, and these eggs roll by the reason of sperm stored there and fertilized. Eventually, when at later stages the sperm roll from the primary location, to the location of vulva, to the location where the ovaries are stored, they got all wrapped up when they go through that passage and come out of vulva. So it forms a process of fertilization actually starts. Thus this opening which regulates it is called vulva. And at secondary larval stages, vulva develop from the vulval precursor cells. Also at secondary larval stages, anchor cell, a prominent member of this signaling connecting the overlying gonad, also develops. Thus here I will try to write the thing that there is an anchor cell, and there are 6 vulval precursor cells, mostly 6 in number. And there is, we see P6P, P6P, P7P, P7P, P8P. I guess it's not writing properly, but P8P. Here we have P4P, and here it's P3P. It's P5P, I guess. It's sorry, by mistake, I guess. It forms the hypodermis or the tertiary vulva. So 6 vulval precursor cells forms an equivalence zone. Why we say equivalence group? Equivalence group means every P6P, P cells, P6P, let it P7P, P5P, P3P, P4P, P8P are equipotent. All are competent. In developmental biology we know induction and competence. So here the inducer is the anchor cell, and all the P3P to P8P, the zone of equivalence group 6 cells, all are equipotent or competent to form the vulva, the primary vulva in particular. So but there is concentration gradient. So most of it, the lim3 binds to P6P, which form the primary vulva. Lesser concentration bind to P5P, P7P, which form the secondary vulva. and the least of the concentration, least of it secreted by the anchor cell bind to the P3P, P4P, P8P, forming the tertiary vulva. And here one member of the signaling is Lin12. So what's the function of Lin12? Lin12 act as a lateral inhibition in plants, let it be animals, and in also plant. You see in trichome initiation, the cell which form the trichome, which helps in ionic sequestration, mostly the cations, anions, and to sequester it to maintain the ionic homeostasis in case of salinity. So the cell which form the trichome, it inhibit the neighboring cell from forming it. Thus it's a very specific signaling. Likewise, Lin12 take part in the lateral inhibition. Lin12, these are homologue of Notch protein. Is a receptor, and most of it express in these cells P6P, and it inhibit P5P and P7P for bilateral inhibition. So it will not take the primary fate. So Lin12 participate in the signaling and helps in lateral inhibition. Here you can see that anchor cell is secreting the Lin3, mostly paracrine factor, which bind most of it to P6P, which form the primary vulva. And here Lin12 express on the lateral side of P6P laterally and inhibit the P7P from having the primary fate. Thus it form the secondary. Lateral inhibition is seen. Similarly, P6P also by Lin12 signaling inhibit the P5 cell. Thus it forms the lateral secondary fate. It take up the secondary fate, and those which are by default receiving the lesser amount of anchor cell form P3P, P4P, P8P, and those are the hypodermis from this diagram. And here while how the question comes from where these anchor cell and vulval precursor cells develop. So their developmental biology is all about one concept is inductance and competence. Some cells induce, some have the competence to receive that signaling, and specification and determination is another concept. At a particular stage, some cell express their mRNA and become determined towards a fate that we have to form anchor cell. It will form uterine vulval precursor cell. So up to secondary larval stages, cells are specified. Then cells get their fate fixed or determination. They move towards the determination. During a particular time at larval development, in wild type C. elegans, two adjacent cells interact with each other, Z1PP, Z4PP, each having a equal competence to form anchor cell and uterine vulval precursor cell. So here you see at the embryonic stage, both random cells near neighboring cells come together. They both have equal amount of ligand and receptor. It means both have the equal potential to form notch, and we have the delta and notch pathway. They can take part, and both have equal potential to form anchor cell, all the Lin12, or it can form the vulval precursor cells, which have more of the Lin12, more of the receptor. So by chance event, some cell like this cell here develops more of the anchor cell properties, develop more of the ligand, or we in case of mRNA we say lag to mRNA, homologue to delta, and some have the Lin12 mRNA which express more of the receptor. Thus having the more of the lag2 forms the anchor cell, and more of the Lin12 form the vulval uterine precursor cells. Thus the cells are specified through this random chance event, and at a critical point one cell start expressing more of the lag2 signaling, inducing other to express more of the Lin12 receptor. Thus a signaling cell which secrete the signal or secrete the hormone, it becomes the anchor cell, and the receiving end, the receiver which have the more of the Lin12 receptors, they form the vulval uterine cells. So this was the overall concept regarding the C. elegans vulva development, and we will be in next lecture, in next video I will be discussing some of the questions regarding it. Thus I think it's a useful topic from CSR point of view, and if you are preparing, that's a very scoring unit, as well as if you are aiming to read it for your semester. Thus I feel that everyone should be able to score it, and as I have some issues with my pointer, anyhow, it's thank you for listening to it, and I hope it will be helpful to you.